Okay. So all of you can see the screen, I hope. Okay. So as we were discussing yesterday, that why is it that there is so much of diversity in the natural world among living organisms? That has been a question that has been bothering humans for a very, very long time. And scientists have been theorizing about it for very long time, since Aristotle and Plato, and actually much before that. Almost all religion, as I said, begins with a creation myth. So then, in modern times, Darwin's ideas got a lot of traction, and there, the major problem with Darwin's ideas was inheritance, which was such a critical part in the whole thing. He did not have a mechanism for inheritance, and this thing finally gets resolved with the rediscovery of Mendel's works in 1990, in 1900, actually in 1900 exactly. Now, once the work of Mendel was discovered and people realized that inheritance is particulate, then a very bitter controversy arose in the field of genetics amongst two camps, which who were called biometricians and Mendelians. So remember, Mendel was showing that traits are discrete: tall plant, short plant, round pod versus wrinkly pod, and so on. Now, people were studying traits in organisms for a very, very long time. And most of the traits that they were studying, so things like height or weight or other BMI and so on, all those traits, they were of a continuous nature. On the other hand, the traits that Mendel talked about, they were of a discrete nature. Therefore, there was a major controversy wherein one side, the so-called biometrician side, championed by people like Walter Weldon and Carl Pearson, they claimed that since the mechanism of inheritance that Mendel's stuff is talking about is discrete, that mechanism cannot possibly account for the evolution of continuous traits like height and weight. Therefore, their understanding was that maybe there is not one but two kinds of inheritance happening, and these continuous traits, they are actually getting inherited by some other mechanism. Now, on the other side, where equally famous people like William Bateson and Hugo de Vries. Hugo de Vries, of course, was one of the people who rediscovered Mendel's papers. And their point was that even if there are two ways in which inheritance is happening, even then, the degree of variation that you find in continuous traits is actually very less. And therefore, that degree of variation, they said, is something that is unlikely to lead to any major changes, and therefore these kind of evolution of continuous traits, they are not sufficient to explain the kind of diversity that we find around us. So continuous traits by the very fact that they have less variation are not important for evolution. Now this debate raged for about 15 16 15 to 18 years, and those days people were let's say not as oak as they are these days. So therefore when they debated it with passion, so things became very ugly, very dirty, and people basically refuse to talk to each other. People refuse to call each other all kinds of names, blah blah blah. This entire controversy was resolved in 1918 by this man called Ronald Aylmer Fisher, when he showed that if you assume that continuous traits are actually coded by a very large number of genes, each one, or factors in that context, each one of which actually produces only a small amount of effect, and then the whole thing simply gets added up over the stuff, the effects of individual. Then he showed that that kind of a system can actually still show what looks like continuous variation, even though the underlying cause is discrete. So, in the process of doing that, in the process of resolving this biometrician Mendelian controversy, he actually ended up putting evolution on a very firm footing, because now the missing puzzle of inheritance was finally resolved. Incidentally, this particular paper, 1918 paper, this paper is also very famous because this is the one where he ends up also giving a bunch of properties of a quantity called variance, which is the square of the standard deviation in statistics. And therefore, this one paper is unique, as in the sense that it is the starting point of not one but two fields: experimental design in statistics on one hand, and the field of quantitative genetics on another. So once this resolution was achieved, then actually things became pretty interesting. So 1918 to 1950s was almost the golden period in the field of evolutionary biology. A large number of very interesting people were working, and they were essentially tackling very, very big problems. And therefore, there are several names over here. I'm just, choosing six of the really big ones: R.A. Fisher, Sewall Wright, Stebbins, Dobzhansky. So Dobzhansky is the person who gave that quote about, nothing in biology makes sense except in the light of evolution. Meyer was the person who ended up giving that biological species concept, the one that we discussed yesterday, and in the process ended up creating a massive amount of confusion in biology. J.B.S. Halden was a British evolutionary biologist, extremely interesting person. He actually ended up coming to India and lived most of the later half of his life in this country, and in the process played a massive role in influencing Indian biology. He ended up bringing Journal of Genetics, which is the oldest journal in genetics on this planet, to India. So I don't know if this is a fun fact. So the oldest genetics journal of the world actually is published from India, by the Indian Academy of Sciences, Bangalore. So recently there has been a very nice biography of JBS Halden written by a guy called Samanth Subramanian. This book is making waves all over the world. I think it has entered into the top 100 list by New York Times or one of those people. I mean, given that this is about a guy who died, almost 70, 80 years back, a scientist, hardly known outside the world of genetics, that's a very big achievement. Very interesting person, very interesting. So anyway, the point is that all these people made massive contributions to the field of evolutionary biology, such that by around 50s, okay, the first 3-4 years of the 1950s, the need was felt that now all these contributions essentially need to be put together, and the core structure of evolutionary theory should be synthesized. And this core structure of evolutionary theory, this basically gets synthesized in 5 or 6 books that gets written in between 1950 to 1955. And this structure, these central tenets, is what in evolutionary biology known as the modern synthesis. This modern synthesis is typically what is taught in schools and colleges, and this is the so-called canonical version of the theory. So what I'll do is I'll spend about 20-25 minutes in discussing what this crystallization is, and at the same point I will also give you guys some updates about what are the things, what else do we know now, okay, what the people who led to modern synthesis, they didn't have the benefit of all that knowledge some 70 years back. So roughly speaking, this is what the structure of the theory is. So if you guys remember this stuff, basic evolution may you guys will never have any issue. Okay. So what it says is that any population will contain genetic variation, and this genetic variation, it can have only two sources. It can either arise by random mutations, or in sexually reproducing species, it can arise by recombination. So this is where modern synthesis starts. However, over the last 70 years, or actually the last 30-40 years in more of it, we now know that there are other sources of genetic variation. So for example, there is epigenetic variation, about which we are going to talk. You can also have variation coming from by horizontal gene transfer from other organisms, other species, sometimes other kingdom altogether. All those are also legit sources of variation on which evolution or natural selection can act. Then it says that populations evolve. Okay. So what is evolution meant over here? So the evolution is synonymous to changes in gene frequency, and this change in gene frequency is brought about by three forces. Okay. Random genetic drift, gene flow is migration, and natural selection. Okay. Now this is a peculiar way of doing it, because remember, we started by saying that evolution is trying to explain how trait distributions are changing. However, by the time we are coming to modern synthesis, we see that it is not talking about trait distribution anymore. It is talking about the distribution of genes or alleles. Okay. Now, this is done because you have this underlying assumption over here that if there is going to be a change at the genetic level, then that will be reflected at the phenotypic level. Okay. In other words, there is a strong 1:1 mapping, or a relatively simple mapping at least, between the gene distribution and the phenotypic distribution. One of the things that we have figured out now is that is not necessarily the case. Okay. In many cases nowadays, we understand that a phenotypic distribution may not have anything to do with the underlying genotypic distribution. Sometimes it's a 1:many and many:1 kind of mapping is possible. So these two other things that I'm talking about over here. Remember, yesterday we said that Darwin was saying that natural selection is the primary force, but modern synthesis explicitly recognizes that two other things are very important. One, what it calls as random genetic drift. So in genetic drift, what happens is that when you are going from one generation to another, then there is a kind of sampling that is happening. Why a sampling is happening? Because remember, each individual, if you are talking of a sexual population, each individual is capable of producing millions of gametes if it's a male, or fairly large number of gametes, a few hundreds or maybe even thousands if it's a female. So of all these gametes that it is capable of producing, only a very few will get combined with each other, and then that zygote that is formed, from zygote all the way to an adult, there are several other stochastic things that are happening. Okay, it might end up dying simply because it drowned in a pond, or just by mistake some guys ended up burning themselves in fire or getting caught by a predator, and so on and so on. Therefore, when you are going from the trait distribution or frequency distribution, gene frequency distribution, whatever, in one generation to the next, then there is absolutely no guarantee that you are going to be able to predict the offspring generation distribution exactly from the parent generation distribution. Okay, and this is exacerbated by the fact that, I mean, even if you can do it mathematically, even if you can do it in the sense of an expectation, in real life, since all population sizes are finite in size, what you will get is a realization of those probabilities. Okay, so going from one generation to another, even if there is no natural selection happening, even then the trait distribution can end up changing a bit. And since any change in frequency is being talked about as evolution, therefore one can say that that itself can lead to a change in the composition and therefore evolution. So these kind of changes, where the change has nothing to do with the adaptedness of the organism, the change has nothing to do with its ability to, how many babies it can produce, those kind of changes are the ones that we say are happening due to random genetic drift. So this is something that we are going to talk about in great detail in one of the subsequent lectures. But as of now, just know this much, that this is gene frequency changes which are happening without any reference to the fitness, to how many babies those organisms can produce. Gene flow is simply migration. You have a population, a bunch of guys from some other place comes and intermingles with this population. Without doing anything, the gene frequency has changed, and by that definition, my evolution has happened. And of course, natural selection we discussed yesterday. So these are the major forces by which evolution can happen. Now, this is the very big one. Most adaptive genetic variation have individually slight phenotypic effects, which means that phenotypic changes over time are gradual. Okay. Now, so basically, this is essentially taking the position of the biometrician, that continuous traits are the ones which are most important. You don't really need to think about discrete traits. Now, this is something that, okay, I don't know why exactly this came about. This is probably, trying to remain true to the original, conceptualization by Lyell, by Darwin based on Lyell. Probably there were some studies in the initial phase, 1950s, and prior to that, which probably led to this conclusion. But today we definitely know that this is not true. There can be adaptive genetic variation, which can happen by changes which are definitely not slight in terms of their phenotypic effects. Large-scale phenotypic changes can lead to adaptive genetic variation. And then it says that diversification at the level of the taxa is happening by a process which it is calling speciation, and this speciation process entails the gradual evolution of reproductive isolation. Okay, so what it is saying is that when, okay, this gradual evolution part is coming because of what has been said over here, that the phenotypic changes have to be gradual. What it is saying is that these small phenotypic changes are going to accumulate over time, and when they accumulate over time, then finally they lead to a state where two populations cannot exchange genes anymore. That is when we say reproductive isolation has happened, and that is when we say that a speciation event has happened. So what the modern synthesis is doing is, it is not only going away from explaining phenotypic distribution, it is also subscribing to a particular species definition. It is going away from the Darwinian species concept, and it is wholeheartedly embracing the biological species concept. However, note the stuff over here. This thing, okay, that if you have a lot of phenotypic changes happening, then that will lead to reproductive isolation. This is something that is not at all a logical conclusion. This one is actually more of an article of faith that if lot of changes happen, then at some point or the other they will fail to exchange genes. That's it. That's the belief. There is no need, there is no necessity for such an event to happen. And what it says is these processes, the diversification through speciation, reproductive isolation, all these processes continued for sufficiently long will give rise to changes of such great magnitude such that you talk about higher taxonomic levels like genus, family, order, class, and all those things. Okay. Now, again, this and this, both you can see are actually trying to stay true to Darwin's original conceptualization, and yet both of them are actually articles of faith, because it is not at all clear that if you see what's the major issue with okay, I don't see it's an issue, but what's the major disconnect between microevolution and macroevolution? Microevolution is what Darwin's conceptualization is trying to explain. All it is saying is that if there is a trait, there is variation in that trait, then that variation pattern is going to change over time. That's it. However, think about it. It is at no place talking about the origin of a new trait. And yet, when you have higher order taxonomic differences, then most of the time that is based on the origin of a new trait. Okay, that is based on here is an organ which this entire group doesn't have. Okay, mammary glands, for example. A fish doesn't have a mammary gland. A reptile doesn't have a mammary gland. Okay, and therefore mammals. Now there is absolutely nothing in Darwin's theory, and therefore there is also nothing, okay, therefore is the wrong word, and there is also nothing in entire modern synthesis which talks about the evolution of new traits. Okay, all it says is that it is possible to take an existing trait and modify it sufficiently so that somewhere it will look like a new trait. Okay, and that is why it takes the, let's say, the vertebral limb structure, the limb pattern, and then shows that how changing that pattern can lead to everything, from the leg of a amphibian to the wing of a bird. But that's not the kind of change we are talking about over here. We are talking about completely new stuffs that did not exist earlier but has now started existing. Those kind of things cannot be explained in this entire conceptualization. Okay, and that is why this has always been a controversy, that this doesn't logically follow. It looks like an article of faith. And today people are slowly coming to the conclusion that novel traits can perhaps arise rather fast, and it can, it's not necessary that it will lead to reproductive isolation, but it can possibly, this designation of higher taxonomic levels, etc., those are also probably possible much faster than what modern synthesis talks about. Okay, so what we talked about here was quite a bit of stuff. So before going forward, I would like to ask you guys if you have any question till this point, because what we talked about over here was very critical. This is like the core of the subject.
Sir, I have a question.
हाँ बोलो.
So, with respect to the higher taxa evolution,
Hmm.
the synthesis or the udbhav of a new organ itself
Hmm.
is, can we compare it to, like, how in Drosophila certain mutations can cause, instead of antennae, legs to grow from the head and things of that sort?
Those are the kind of stuffs that will definitely, qualify as a major mutation. Okay. However, there are, or a major change. However, there are all kinds of issues over there. The major issue is that people claim that if you have such major changes, then those guys are not going to be viable. Okay.
I
So
see.
mutations of large effect size are typically going to lead to the death of the organism. So that's the secondary thing that people end up saying. And you can see why that is so, right? If you suddenly have, legs coming out of your head, then okay, fine, you can have the stuff over there, but obviously the organism will not be able to walk on those legs, right? And those legs are going to cause all kinds of other troubles for the organism. So how do you expect that thing to survive? How do you expect blood vessels, to find their way into such legs? So it's a massive debate, and people are doing lots of experiments on those. The entire field of evo has essentially come about to answer some of these kinds of questions. um, yeah, I think without going into details, it's difficult for me to say more on that.
All right, sir. Thank you.
Hmm.
So
हाँ, बोलो.
in point 2, you also talked about gene flow, correct? Could you
I
please repeat
gene,
that?
gene flow is migration.
Okay, it's just migration. Okay.
Yeah, that's the technical population genetics term for migration. That's anything else?
So also, you said that changes in the gene frequency need not necessarily be related to changes in the phenotypic frequency.
Yes.
I mean, how?
Yes.
How? How so?
See, the point is that here the underlying assumption is that each gene maps to a fitness, right? But we know that several genes sometimes lead to the same phenotype. So even if there is a underlying change in the frequency of, let's say, those 3 or 4 redundant genes, at the phenotypic level, you will not feel that because all of them are leading to the same phenotype.
Okay,
You
sir,
get
makes
the
sense.
point?
Yeah, I
Hmm.
get it. Thanks.
Anything else? Okay. So then, now please note one thing over here, which is this date. Okay. This thing is happening in the first half of the 50s, which means, remember, the Watson and Crick's DNA paper, DNA structure paper is 1953, right? So that means that all this stuff that we are talking about over here, all this more or less entirely predates the massive amount of information that we have gathered from molecular biology in the last 60 70 years, right? And molecular biology is not the only field. Okay, we have made very massive strides in understanding how, for example, cells communicate with each other. So that's what this one is about. Uh, so that's what this one is about. Then one subject in which our understanding has grown by leaps and bounds, also due to some major technical advancements, is developmental biology. So now we have a much greater understanding of the kind of things that I think Vikram was talking about, which is, you take a gene, you express it somewhere else, and you get a very different pattern altogether. So all those stuffs have come about. And finally, over the last 70 years or so, we have understood a lot about how the environment around us functions, how environmental features end up affecting pretty much everything that is there in the body of an organism, starting from its gene, the expression of its genes to everything. Therefore, one of the questions that people have been asking for the last, say, 10 to 20 years is that, hey, there is so much of new knowledge that is available. Shouldn't we rethink the theory of evolution in the light of whatever we have understood in the last 6-7 decades? And if we do that, then one assumes that the theory of evolution is going to, it, in the best case scenario, it will expand slightly, because all these new things will have to be incorporated somewhere or the other. And in the worst case scenario, we'll have to completely junk whatever I showed you guys as modern synthesis, and we have to come up with a completely new theory itself. So that notion, re-examining evolution in the light of advancements in the last 6 to 7 decades, is what is known as extended evolutionary synthesis. And this, again, like everything else in evolution, evolutionary biologists are the ultimate argumentative people. So this has basically divided the community into two parts, and there are lots of people who are of the opinion that, look, the fundamentals of the theory are still very strong, so all these new things will at max allow us, or at max require us to put some kind of an addendum here, or make a more nuanced statement there, etc., etc. But the structure of the theory, as I outlined, that structure is not going to change. That structure is going to be very, very solid. On the other hand, there are a few people, and as usual, these are very vocal people, who are shouting from the top of their roof that no, this is a mistake. Entire evolutionary theory, the structure is rotten, and we have to basically completely change how we think about evolution based on all these new things. Okay, and there is a lot of back and forth that keeps on happening between these two groups. We ourselves have contributed to these back and forths, and we had ended up solidly bashing the other side in a rather how do I say, controversial way. But anyway, so this is what it is about. Even if you don't think about the controversy, what you have to realize is that there are lots of people, the vast majority of people, who are actually not taking either sides. They are simply putting their head down and working and trying to see for themselves which is the side that is more important. I mean, does evolutionary theory simply require some edits, or does it require a complete restructuring? Over the last 15-20 years, there has been zero indication that the theory needs to be completely abandoned. Okay, there has been no major paradigm shifts, and frankly speaking, with more and more progressing time, it looks like at least the existing knowledge will not lead to a paradigm shift. If some new knowledge comes about, that's a different ballgame altogether. But people are, at least some of us are reasonably sure that nothing will be required for the existing knowledge of the last 70 years. However, it is forcing us to rethink some of our concepts in basic biology. So what I'll do is I'll quickly discuss 2 or 3 of those concepts in the context of this class, and leaving out some of the more controversial ones. Yeah, because there is more meat in what I'm discussing compared to all those controversies. Anyway, this book, Richard Dawkins, The Selfish Gene. How many of you have heard about it? Don't write it in the chat box because I can't see the chat box. Just say some yes or something.
I have.
Almost everyone.
I have.
How many of you have read it actually?
I have read half. I'm halfway through.
I guess this was mentioned in HSS course.
Any mentioning is fine, but how many of you have actually read it? See, the point is everybody knows about the existence of the Bible, right? How many have read the Bible? And ditto for Gita and Quran and everything else. So similarly, this book is almost supposed to be the Bible in evolutionary biology. I want to know how many have read the Bible. Somebody said half. Anybody full?
Oh, yes, I've read it.
Okay. Okay. So, what is the main thing that Dawkins is trying to say in this book?
That genes are the fundamental entity that play a role in selection.
That is one thing. Genes are the fundamental entities, and therefore, by being the fundamental entities on which selection operates, according to Dawkins, they are the most important thing in the evolution, right? So, the way Dawkins puts it is that it's essentially life is just a continuation of genes over time, and this entire phenotype, this entire body that we have, is just a vehicle which the genes manipulate to ensure that they, remain forever. They pass from over time, right? That's pretty much what he says in this book.
Yes, he mentions the body is a survival machine and gene is something that has its consciousness.
Correct. Exactly. So it's like a robot, right? Survival machine robot, whatever. Okay. So the major point, obviously, in this whole thing is that he says that the features of an organism are almost entirely determined by its genes. Okay. And this conceptualization is something that is by no means unique to Dawkins. Most people in the field of molecular biology actually very, very strongly believe in this conceptualization, which is why much of molecular biology today, unfortunately, has essentially ended up becoming, find the gene for whatever phenomena you are observing. And this is where I get into a lot of controversy with my colleagues over here. Most of them have actually, some of them have told me point blank that if you are not searching for the gene, then what are you doing? Okay. So in the eloquent words of one of them, my study begins where your study ends. Okay. My entire reason for existence begins when I start looking for what is the underlying gene. So there are these superb comments, like amoeba and elephant. What's the difference? If you tell me what is the gene underlying all the functions in the amoeba and the elephant, I'll tell you exactly how the amoeba behaves and how the elephant behaves. Now, obviously, that is something that is probably not correct. And because it is not entirely correct, therefore, that has consequences for how evolution works. So the main implication of thinking about what is known as the gene sci point of view of biology is that the role of the environment is merely as an agent of selection. Environment is just creating the context in which the genes are expressing and doing their function, and then it is simply the environment is sorting out the through selection which frequency will increase and which will not. That's about it. However, over the last several years, we have come to understand that that is possibly too simplistic a conceptualization. So, okay. Do you guys know what the epigenome is?
Yes, sir.
What is the epigenome?
So I'd say I think it should be like the spatiotemporal variation in the expression of genes rather than the genetic composition itself.
No, technically speaking, that is not what it is. Okay. Anybody else has a better understanding or whatever, better definition?
Epigenome, as much as I understand it, is all the control that occurs above the genetic level, in the levels of protein, or maybe some sort of manipulations that happen at the chromosomal levels, or arrangement of chromosomal levels, which control the expression of certain types of phenotypes.
Okay. Controls the expression of phenotypes, yes. Controls, I mean, that happens through proteins, yes. But typically the term epigenome is reserved for a slightly more constrained set of things. Okay. I have only 5 minutes. Okay, we'll stop over here. Okay, we'll stop over here, and we stop essentially with this thing, that according to this Dawkinsian gene-sci view of the world, features of an organism are driven by the genes, and environment is merely an agent of selection. And from this, we are going to see how the concept of epigenome, epigenetics, etc., all that ends up overturning that, and then we'll talk about what's the implication of that for evolution. Okay, we'll start that tomorrow. I need to show you guys a video, but I realized that if I play that video from there, then you guys won't be able to see it. In the sense, you'll be seeing it, you won't be able to hear the commentary, which is important. Okay, so we'll stop this class over here, and tomorrow we are going to continue with epigenome and what that means in the context of extended evolutionary cycle. Okay. Yeah, okay. So I can take one or two quick questions.
Sir,
Okay.
yeah. So, why is the idea of the epigenome at odds with the idea that the selection operates at the genetic level? Like,
No, I didn't say that. I didn't say that at all. I said that the consequence of the gene-sci view of evolution is that the environment merely acts as an agent of selection. Okay, the environment doesn't play a role in the generation of the variation itself. And
okay.
so that is the tenet that gets overturned.
Yeah, correct. Thanks, sir.
Yeah.
So with respect to trying to find a gene for every the argument that you mentioned, isn't it looking at the same question from just two different points of view? So they're alternate viewpoints for explaining the same phenomena. So why do they have to be at odds with each other?
No, they are not at odds by any means directly. Okay, the problem happens is that, remember, genes are at a different structural level of organization. Okay, and the phenotype is at a different structural level of organization. Then you have the organism, and then you have the population, and so on. The issue is that when you are talking in terms of what interests you, okay, some people are of the view that until and unless you connect it to the genes, then the whole thing doesn't become interesting. And as, okay, I don't know if I can talk about it in the context of this lecture, but most interesting traits that we study, most of them, they are polygenic. Okay, and figuring out a gene for them is close to impossible. At least with the modern techniques that we have, it is close to impossible. And I mean, this is a problem that is very well known to people who do this kind of work, that the entire toolbox of genetics or microbiology, whatever you call it, that is actually geared towards finding those genes or those pathways which are very simple. You make one alteration and you have a big phenotypic change, very few causal links going from that gene to that phenotype. Those are the only kinds of things that they can catch. If you have a more nuanced phenomena where the underlying phenotype is determined by multiple genes and these are interacting in all kinds of complicated ways, then just figuring out which are the genes involved is a very, very, very uphill task with the present kinds of techniques that are available in the toolbox.
But sir, doesn't a transcription network do exactly that?
कुछ नहीं होता. See, that's the whole problem. It only works for the very simple cases. For the complicated cases, it's almost impossible to figure out. तुम कुछ भी कर लो. I mean, we can discuss this separately, but it's actually a very, very difficult thing to figure out. And therefore,
Sir,
just 1 second. I think it's 45. So let me stop the recording so that people who want to leave can leave.